Hello, my name's Dr. Freya Harrison and I'd like to tell you a little bit about what we're doing in my lab at Warwick. We're particularly interested in why bacteria can cause very antibiotic resistant, very long-lived, hard to treat lung infections in people who have the genetic condition cystic fibrosis. Now, cystic fibrosis is one of the most common genetic disorders among people of European descent, and about 10,000 people in the UK are living with this condition. What it is, it's an imbalance in the way your body moves salt and mucus around. And one upshot of this is that the lungs of people with cystic fibrosis produce very thick, sticky mucus that's just really, really hard to cough up. And what that means is that any bacteria that go into the lungs, so the bacteria that normally live in your nose and mouth, or bacteria from the outside environment, go in and can't get out. And so people get very biodiverse infections of different bacteria living in the lungs. And sadly, this means that most people with cystic fibrosis die of respiratory failure. And these infections start in infancy, but they really become a problem for people as they hit their teens and early 20s. What's really and very fascinating about cystic fibrosis infections is that we don't understand why they're so hard to treat, how they keep one step ahead of your body's immune defences and one step ahead of medical treatment, and where we might find a weak spot to treat these infections. One reason for that is we don't have a good laboratory model for studying these infections. In my lab, we're building a new lab model of cystic fibrosis infection. We take lung tissue from pigs that have been killed for meat, we infect it with the bacteria that cause infections in cystic fibrosis, and we add this infected tissue to artificial cystic fibrosis mucus that is made in the lab and is chemically very similar to real cystic fibrosis mucus in the lungs. And using this system, we can study how bacteria grow, evolve, interact with each other in the lungs, and how they respond to existing or potentially new medical interventions. One thing I really like about working with this model isn't just that it tells us a lot about cystic fibrosis, but that we are potentially reducing the use of animals in research. Because we use a byproduct from the meat industry, we're not requiring the use of live animals to study infection, we're not requiring the slaughter of any animals to use our model. So we have a fabulously ethical model, and I hope that by encouraging colleagues in Warwick and around the world to use this in their research to study cystic fibrosis and other infection contexts, we can help to reduce the use of animals in research. Fundamentally, anybody who works on cystic fibrosis lung infection or on long-lived infections in other clinical contexts will tell you we don't really understand what's going on inside the patient in these infections. It's a black box. And what my colleagues and I hope is that through our research we'll be able to open that black box and look inside.
Hi, my name is Dr. Jose Gutierrez Marcos, and I'm going to talk about memory in plants. Contrary to animals, plants cannot escape from stressful environmental conditions, such as high temperature or lack of water. Despite these limitations, plants have successfully colonized all parts of our planet. This is thought to be possible due to the unique developmental flexibility of plants to adapt to different environments. For example, plants previously exposed to lack of water produce offspring better adapted to drought. Although plants do not have a brain, they have the capacity to record this information in their DNA by chemical modifications of the genome and transmit this information to the offspring. Environmental signals or stress regulate enzymes that modify the DNA by methylation, leading to changes in the way DNA is packaged, and this affects the way genes are turned on or off. This chemical information can be copied by enzymes and transmitted to gametes and therefore to offspring. Because rapid changes in climate conditions affect plant growth and crop yield, we are looking into ways to manipulate plant memory to increase productivity.
Hello, my name is David Roper. I'm a biochemist and I do research in antibiotic resistance. So my interest in science came from a love of both biology and chemistry at school, so it seemed natural to me to focus on biochemistry, which is at the interface of these subjects and I'm fascinated by how life happens at the molecular level. I mostly teach on the biochemistry degree course, and that influences both the range and diversity of my teaching. The research in my lab has always been driven by the biomedical need to treat antibiotic-resistant infections and address the problem of antimicrobial resistance, AMR. Pretty much everyone in the Western world has been treated with an antibiotic at some point in their life, and pretty much every aspect of healthcare is dependent upon these drugs. So it's not just the immediacy of a life-threatening infection you have to consider. Some aspects of surgery and ongoing patient care, from diabetes in the young to joint replacements in the elderly, will be really difficult in the future if we don't get to grips with this issue. The problem of antimicrobial resistance is not just a scientific or medical issue. It actually impacts many other areas of human activity, from food production to the environment to society and economics. For example, developed countries are at threat from the poor use of antibiotics that promotes the development of antibiotic-resistant infections in individual patients, whole communities, and even farm animals. At another level, many people in poor countries have limited or no access to these drugs, and when they do, they can be poorly administered. Most people assume that the drug companies are on top of this problem, but there are very few drugs that are coming through the pipeline, and these companies are very reluctant to invest further as they don't get enough return on investment compared to heart disease or cancer, for example. So the research in my lab goes to the heart of the issue. How do these bacteria become resistant to these drugs in the first place, and how do we find new ones? Our research focuses on how bacteria make their cell walls, because this is an Achilles heel in bacterial metabolism. Whilst we have known for decades the basis of how bacteria make their cell walls, how this is coordinated and how this is related to how these drugs work is largely unknown. We now understand how we can target more than one protein at once, which is how penicillin-based drugs work. But we can also target the substrates for these enzymes in the first place, and that may be the future of the next generation of antimicrobial drugs. So in conducting this research, we're discovering the fundamental science surrounding antibiotic resistance and also developing new knowledge that may help in the discovery and development of future drugs.